Hello everyone, this is SMJ and today I'm going to give a presentation about the paper which is titled Quorum Catching Bacteria Isolate from Industrial Wastewater Sludge to Control Membrane Biofouling. Order of presentation is here. First, I am going to give an introduction, the background and methodology and result of them, and main point of this paper, and finally I will wrap up with the further research. First is about introduction. Before I start, I want to give a question. Do you know that animals or plants can communicate with each other? Then how about the microorganisms? Well, they actually do the communicate. And furthermore, sometimes it become the problems. MBR is a membrane bioreactor. This is the process combine the biological treatment and membrane filtration. It has many good points with the MBR, like here, but there is a main drawbacks called membrane fouling. Membrane fouling is the clogging with the membrane pores like here, and is related to biofilm formation. And biofilm formation is also related to quorum sensing, which is a communication for the microorganism to control their group behavior. So to inhibit it, the strategy called quorum counting QQ has invited. And objective of this paper is finding new endogenous QQ bacteria from an ambient plant which treat the industrial wastewater containing the toxicant called TAMA. Second is methodology. First methodology is isolation. Isolation is a method to spotting and catching the specific microorganism with the special functioning. In this paper, they sampled from the wastewater treatment plant in Korea, and they used the chemicals which is lower in the quorum sensing, and as a language, I will call it language chemicals. So they use the language chemicals for the isolation method. And after isolation, they found 10 of new microorganisms, new QQ bacteria, and with the RNA sequencing, RNA analysis, they found the name of them like here. And next methodology is about QQ activity test. After we find the new QQ bacteria, we need to know how well they actually do the quorum catching. It is about that. So they culture new QQ bacteria with the language chemical and also language chemical and toxicant TAMA. And they measure the luminescence, the light of the reporter strain called A136. A136 is a microorganism which emits the blue light like here with the existence of the language chemicals like here. Next is the result of them. First is a actual picture of isolated microorganism. This is the picture of SDC A8 and SDC U1. Among the 10 microorganisms they isolated, they did the best performance in the quorum counting. And this is a graph about the QQ activity test. As you can see here, x-axis is time and y-axis is a concentration of the language chemical. So this is the black line, which is a contour, and as you can see, they did not degrade the language chemical at all. But and while the red line and blue line sharply decrease, which means that the QQ bacteria they isolated is well degraded the language chemicals. And this is a bar chart. Those are the many types of the language chemicals, and this is a degradation rate of them. As you can see, the orange bar is sometimes higher than blue bar, but in general, the blue bar is higher, which means that, the orange bar is mostly higher than blue bar, and the orange bar means the acidity ion, which means that acidity ion it's better to degrade with language chemical than acidity A8. Last graph is about last graph is same experiment but with the toxicant tama. This is the acidity A8, U1 A8 and other acidity D40, but in here also acidity U1 well degraded the language chemical with or without the tama, the toxicant. So what is the main point of this paper? The main point is that they found a new QQ bacteria from the ambient plant, ambient sewage, which treat the industrial wastewater containing tama. And among them, Bacillus acidity U1 and Bacillus acidity A8 show the most highest QQ activity with or without tama. And their higher QQ characteristic and growth rate mean that they are the promising candidates for mitigated biofouling in ambient. This is the final part of my presentation. First, why this paper is important? For this field, actually, the QQ bacteria found this is limit. There are not that many, so more is needed for the further research for QQ ambient or quorum catching itself. And for me, personally, this paper is actually from my professors and our laboratories. So this is so meaningful because I also want to do research about similar topic with this paper. And further research, this is also for me. I want to find at least one new QQ microorganism during my master degrees. And also, I want to combine the acidity one, which is found in QQ bacteria in this paper, and other bacteria before founded. So I want to combine those microorganisms. So thank you for listening for my presentation. Thank you.